In 1907, scientist Joseph Marx described the inflammation of major nerves in laying chickens. He believed the paralysis to be caused by an infectious viral agent. This was the first description of what would later be termed Marx disease. Over the years, several other scientists like Erwin Pappenheimer, Tony Churchill, and Peter Biggs also made discoveries that provided broader information about Marx disease. So, what is Marx disease? It is useful to note that Marx disease is caused by a virus called Marx disease virus. Marx disease virus is a cell-associated herpes virus which results in tumor formation, immunosuppression, and neurological disorders in chickens. Some symptoms of this disease include paralysis, formation of tumor, and changes in eye condition. My mentor, Dr. Mark Purcells, was able to obtain 30 FDA cards from Nigeria with blood and tumor samples infected with Marx disease virus. The Nigerian poultry industry is one of the most commercial subsectors of Nigerian agriculture. The intensity of poultry rearing keeps on increasing, resulting in reemergence of Marx disease. Marx disease has a huge economic impact. First, because of continuing losses due to the disease, and second, because of the cost of vaccination. So, for my project, I worked on the purification of DNA for amplifying genes that are divergent among Nigerian field strains of Marx disease virus. A mouthful, I know, but the purpose of this project was to enable genetic analysis and expression vector cloning of specific Marx disease virus genes. Several genes unique to Marx disease virus have been identified. Among these, the MEC gene is found to have oncogenic expression and is related to Marx disease virulence when they undergo mutation. Noting this, I formed my hypothesis. Based on size and stronger correlation with virulence, MEC gene will be most suitable for amplification in order to study Marx disease virus. The first step in my project was the purification of the DNA samples sent from Nigeria. Because the submitted samples were from international poultries, I had to handle them as if they potentially contained human or animal epizootic pathogens. Consequently, I also performed an RNA treatment step in order to inactivate RNA viruses. After the purification, I had to dilute my DNA yields in order to get them in the 25 to 50 nanogram range. This diluted yield was recorded and marked as the working stock. The next step was the amplification of MEC genes from the DNA yield obtained. To do this, a PCR was carried out using a capacity PCR tube. After this step, the amplified products were visualized under UV light as a single compact band of expected size. The third step was the topo cloning procedure. I made use of the PCR product from the MEC gene amplification for this process. A topo reaction solution was prepared, put in TOP10 bacteria, and spread on an LKX plate.
This is the LKX plate which we use to spread the bacteria.